So thank you all very much for the invitation to come and speak today and present some of the work that my group are doing. It's nice to get out of the lab and get out into the sunshine. So leading on from that introduction, so my research group is primarily interested in understanding the electrical and structural underpinnings of changes in the brain that lead to the panoply of behaviors that individuals can experience during neurodiversity, particularly in Fragile X syndrome. So I'm going to talk to you today about some studies we've done looking at the rodent models of Fragile X syndrome as a kind of test bed to kind of understand circuit function better. So I'm sure I don't need to explain to you all, and the talks that have come before have done a really excellent job of explaining the molecular basis of Fragile X syndrome, particularly the genetic background. But I want to give you a quick kind of update of what aspects of Fragile X syndrome, particularly at the kind of molecular level, that are relevant to our research. So we know that Fragile X syndrome results from depletion of the FMRP protein that's encoded by the FMR1 gene. To the degree at which FMRP is lost differs between individuals, and I'm going to talk today about full mutation, so full loss of FMRP. But this, when we have depletion of FMRP, this leads to an imbalance of cells in the body in their respect to their gene and protein expression. So the amount of protein or gene that individual cells might express changes, and this is one of the reasons we think we get altered brain communication. And it's this altered brain communication that we really think underlies lots of the different behaviors and experiences that individuals with Fragile X syndrome can have. And there's a growing body of evidence that this long-range connections between different parts of the brain are really key to understanding that. Some of you may have experienced this type of clinical study in the past, this particularly electroencephalograms, where a skull cap is placed on an individual and we can record the activity of the brain from individuals. And we know from a number of recent studies that individuals with Fragile X syndrome have differences in how the brain processes activity. And so what I'm showing here are two examples, one from typically developing individuals and some from Fragile X individuals. And what you can see based on the heat map here, so where hot colors equal high activity and cold colors equal low activity, this is the activity of the brain in response to an auditory stimulus. So much like the previous talk talking about sensory reactivity, when an auditory stimulus is given to a typically developing individual, you see activity in the brain regions that correspond to auditory inputs, so those brain areas that process sound. And when this study performed this EEG type analysis on individuals with Fragile X syndrome, what I hope you can see is that the activity in the brain over those areas is much higher, and that's represented by hotter colors. But not only that, but that increase in sensory activity leads to increased activity across the rest of the brain as well. So you get a spread of high activity out from that sensory region across into the rest of the brain. And we think this change in connectivity and this change in activity might be underlying some of the behaviors that individuals experience. So trying to think about, my lab's really interested in how the brain structure leads to function. And so when we think about the brain, and particularly the human brain, all individuals, Fragile X or otherwise, are able to perform incredibly complex tasks. You can talk, you can interact with objects, you breathe, you eat, you go to the bathroom, whatever. And all of these behavioral tasks require multiple areas of the brain to interact and coordinate their activities simultaneously. And in the human brain contains approximately 86 billion neurons. And these neurons form connections, and they might form connections locally within a small area, or they might form connections across the other side of the brain, or with the rest of the body. For example, a motor neuron in the brain ascends from the top of the brain right down to the tip of the toe. So these neurons can be huge, and they can travel vast distances. And they form many, many, many more connections than there are neurons. So there's approximately 140 trillion connections in the brain. For a variety of reasons, most of them ethical, we don't study human brain tissue. We prefer to study preclinical rodent models. So we use the mouse model, and this is not to scale, but the mice contain many fewer neurons than humans. So mice contain approximately 70 million neurons in the brain. But really importantly, they comprise many of the same brain areas, and many of the same connections exist between brain areas. And just to kind of highlight one of those areas that I'm going to talk a bit more about in my talk, one area highlighted in green is an area that we refer to as the hippocampus. And this is a brain region that's really important in remembering where you've been, remembering what's happened in that location you've been in, and allows you to remember that event in the future to inform future behavioral tasks. And importantly for us, the hippocampus is conserved between mice and humans. And what I want to highlight is, in the mouse brain, even though this is not to scale, and realistically we're talking about something about that size, and this brain actually is my head, but this is this is actually, maybe to scale, I don't know, depending on how egotistical I'm feeling. Unfortunately for me, I also have a hippocampus, so I will remember being here today. But what you can see, and what I want to point out, is that the hippocampus in rodents makes up a much larger proportion of the brain volume than it does in humans. And this is due to cortical expansion in humans. And we can see that the cortex here comprises much more of the brain volume than it does in lower mammals. And this is really reflecting our ability to perform much more complex behavioral tasks. I will point out as well, just as a point of reference, to the best of our knowledge, there's no difference in the number of neurons and the structure of the brain in individuals with Fragile X syndrome. So the overall structure is more or less identical to those typically developing. So just to kind of go a little bit more into the hippocampus as a brain structure, I've worked in the hippocampus for approximately 15 years now. I love it. I know every ins and outs. I could talk for hours about it. But what I really want to convey is that the hippocampus encodes this information about where and what we are doing, where we are at this moment in time, what's happened, and how that relates to previous experiences we've had of that. Okay. It's the land train. Excellent. And it informs our decisions that we make in that environment in the future. And this is really important to how we continue to develop behavioral tasks and how we continue to develop ourselves, our sense of self. Importantly for us, experimentally, the hippocampus is common to all mammals. So mice have a hippocampus, humans have a hippocampus. And really importantly for me, I'm a really simple creature. I like things that look simple. It's really easy to identify in the brain. And you can see it in this schematic here. This is the hippocampus highlighted in green is really well defined and exists in a relatively stereotyped manner between human individuals and also rodent individuals. So you can identify it again and again and again, and the same cells exist generally between areas. So why do we use FML1? Why do we use mice as a model for Fragile X syndrome? Well, first of all, as I keep pointing out and banging on about, they're mammalian, so that's good. But really interestingly, they're highly genetically modifiable. So we can change the genes in mice to try to mimic as close as possible the genetic changes that occur in Fragile X syndrome. So we can change the FML1 gene, and we can alter it such that it reproduces FMRP depletion. And the FML1 knockout mice, which for the rest of the talk I'll highlight in red just so it's clear, they present several features that are similar to that of Fragile X individuals, in that they have altered gene and protein expression, they have altered learning and memory, and some of the mice are more prone to seizures, which some of the individuals with Fragile X also experience. So we think that they're, in some respects, a good model of some aspects of Fragile X syndrome. So as I kind of alluded to, my lab is really interested in how we link form to function. So as I alluded to, this is the brain. In the brain, we have these neurons that look a bit like this. And the brain is comprised entirely of these neurons with non-neuronal cells as well. But every neuron has an input region, which I highlight in black, and an output region, which I highlight in red. So these neurons receive tens of thousands of inputs. They integrate them to produce a response, which then gives an output. And then we can actually visualize those individual inputs, and these little spiny protrusions here, each one of those is an input that a neuron receives. So we can, in the lab, image and visualize each stage of humans or brain structure from human or rodent. And importantly, we can measure the electrical activity of those cells and those networks of brain cells at the same time. So here's a recording that I did a few years ago of a population of neurons in the rodent brain, and you can see it's got this spiky structure that looks very much like brain waves. And this is essentially the substrate of brain waves that would be recorded with an electroencephalogram. Neurons themselves produce these responses by spiking, and this is the spiking output of a single neuron. Actually, it was this cell. And you can see that these spikes are the way that a neuron communicates with the next cell. So it spikes, and it tells the next cell to spike. And that's how information is transferred around the circuit. But also we can measure the activity of these individual synaptic contacts. And they look something like this. And they're very small, very transient, and they sort of go blip, 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 and I could watch them for hours. But what I want to really highlight here is that the neurons we see in mice really closely reflect what we see in humans as well. So the overall basic properties of neurons in mouse neurons, and these are some cells that I recorded in the lab a couple of years ago, they really closely resemble human neurons. And these are human neurons recorded under exactly the same conditions as mouse neurons that we did in some tissue that was removed during tumor resection. And you can see that they look pretty identical in terms of their input structure, and also it's a little difficult to see their outputs as well. So how we go about recording the activity of single neurons is really tricky. And it's not something that's done every day, and it's not something that most neuroscience researchers do. And the reason is that neurons are really, 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 really small. So if you imagine your human hair, this is about 50 micrometers wide. You've got thousands of human hairs. An individual neuron in the hippocampus of the mouse, or human, is roughly 5 times smaller than a human hair. To measure the activity of that neuron, we need to stick an electrode, which is a glass pipette, essentially, onto the membrane of that neuron. The glass pipette itself is 1 to 2 microns wide. So you're imagining we're trying to guide a needle, which is 50 times smaller than a human hair, at something that's 2 or 3 times smaller than a human hair. It's sort of prone to error, but it's quite reliable in our hands, and we can really get a great access to the electrical activity of neurons. So why is this all relevant? Well, we can test this between wild type, typically developing mice, and FML1 knockout mice to look at how neurons are responding differently. So here's a response of a neuron in a wild type mouse in the hippocampus, and here I'm doing nothing to it. I'm just leaving it just sitting, doing its own thing. You notice it's not very active. These are neurons that have been removed from the brain, so they don't have ongoing behavior, so they're pretty quiet. But when I give stimulus to this neuron, we can slowly increase the response of the neuron in steps up to a point where it produces spikes. And this is very artificial, very precisely controlled, so we can do it time and time and time again across animals and across developmental stages. But what I want to point out is there's a level where the neuron produces those spikes. And if we take that level across here, and we measure the same thing in an FML1 knockout mouse from the same brain area at the same developmental stage, we get the same response at baseline, not very much going on. We get a response to increasing stimulus, which looks broadly speaking the same. But what you'll see is the spiking comes very much earlier. So these neurons have a much reduced threshold to produce output. So they're able to transduce information really, really rapidly and much more efficiently than wild-type neurons, which in some ways could be a good thing, but in some ways it can also be negative in that if you have typical information coming in, it produces too much response in response to a stimulus. That makes sense. So to confirm that I'm not talking nonsense, we repeated this a bunch of times, and we published this a couple of years ago, during COVID actually. And what you can see in wild-type, typically developing mice, as we increase the input, and the numbers don't really matter here, you increase that input, the neurons increase their spiking output, as shown on the y-axis. But what you'll see is there's a really strong leftward shift in the FML1 knockout neurons, basically implying the overall threshold for their activity is shifted to lower levels. And the analogy I like is, if you imagine a light bulb in the house, you require a certain amount of electricity to light a light bulb. If you then go out and buy and try to change all the light bulbs in your house to low energy versions, you suddenly need a lot less electricity to produce the same lighting. And it's exactly the same in FML1 knockout neurons. They are more responsive to a lower level of activity. But I guess kind of curious neuroscientists to be said, well, what's the reason for this? Is there something about the structure of neurons that has changed that could contribute to that? And we know from lots of work of much more intelligent people than I, that the ability of neurons to produce these spiking responses is mediated by a really small part of the cell known as the axon initial segment. And this sits right between the input part of the cell and the output part of the cell. So we asked, and we can measure this structure, and here's an example that we did a couple of months ago in the lab. This is a human cortical neuron. The neuron itself is labeled in blue. And apologies for those who may have color blindness. I really inadvertently put red and green here for some reason. But the important bit is here. You can see this kind of small little green structure here. That is the axon initial segment of a neuron. And we can measure it reliably, and we can image these under high resolution microscopy again and again and again. So we did that. We took the wild-type mice, same wild-type mice. We measured their axonal initial segments, this structure that encodes integration of this spiking, and we did it in neurons, which are now shown as blue. And we can repeat the same imaging in the FMO1 knockout mice. And the prediction would be, if we have an increase in excitability that's due to this structure, we would see a change in its length. And that's exactly what we see. We see approximately 10 to 20% increase in the structure of the neuron, this structure of the neuron, which corresponds to this activity, telling us that our first finding is pretty much based in something that's structurally different about the neurons. But, and this is where it sort of gets a little bit tricky, is that this axonal initial segment and the firing activity of neurons in this reduced threshold tends to happen in response to changing activity. In that, if neurons increase their responsive activity, and neurons like to remain at this sort of warm, happy, we're firing at a constant level, everything's sort of in this sweet spot of activity. So if the neurons are more excitable, something must have changed to maintain that sweet spot level of activity. And the most obvious thing that it could be is that the neurons themselves receive altered input or reduced inputs to try to kind of return to this warm, fuzzy, happy rate of firing. And so that's what we wanted to ask is, we have this change in excitability, we have this change in this neuronal structure that corresponds to that threshold for excitability. Has something changed in these long-range connections that the neurons receive to account for that? So to do this, I switched up the experiment slightly. I say me, my incredibly talented colleague did this actually. And she placed a stimulating electrode, which is essentially just a little battery attached to two wires, in the pathway that corresponds to these long-range inputs to the hippocampus. And here's the electrode recording this cell here. And when we produce these responses in wild-type mice, we get this large response that is hopefully reasonably clear that the stimulus is producing a clear response in the neurons. When we do exactly the same in the FMO1 knockout mice, this is significantly reduced. And really importantly, these long-range inputs are the exact same inputs that are producing this sense of where we are and where we are within our environment. So essentially, the hippocampus, which is the area coding the memory for where we are, is receiving much lower levels of information that encode where we are. Hence, they compensate up their activity to then try to normalize that function. And so we kind of come back to this diagram here, that we see this increased neuronal output, this spike activity, but we also see a big reduction in this input to the neurons. And basically, the upshot is that this is leading to a balanced response to the neurons. So the neurons are not going into a seizure every time you move into a new room, but that's at the kind of compromise of a loss of information. So you're having a reduction of sensory, or the reduction of spatial information, which is then being kind of normalized out by the circuit. That's all great. It tells us that neurons in a mouse model of Fragile X syndrome are doing something, and that may explain it. But what we really want to know, and we really want to understand, is how do these inputs, which we see as being the main mechanism by which spatial information is transferred, how do they change as we experience new places and new experiences? So, with the idea being that if an individual struggles to learn a new environment, is there a cellular substrate for that impairment? And so, to do that, what we want to do is use a molecular mechanism, or a mechanism we can use in the lab, where we can mimic new memory formation. And the analogy I like to use is, every connection in the brain likes to be activated repetitively. So if you learn to use piano, for example, the first time you use a piano, you just sit there and you clunk keys away and you don't play anything. If you practice and practice and practice and repeatedly play the piano, you get much better at it. And connections in the brain are exactly the same as that. So here's an example. We'll go back to this kind of diagram. Here are some connections in the brain. They produce this sort of response. If we stimulate those responses, you get the same response. So now, but if we kind of use the analogy of practicing the piano a lot, if we repetitively activate that synapse, it becomes much bigger and strengthens. And this is essentially how we believe memories are formed in the brain. So if the ability in rodents the FMO1 knockout rodent model are less able to code spatial information, maybe there's a deficit in their ability to strengthen those inputs. So in wild-type mice, we can produce a response, we do that strengthening paradigm, and we see a bigger response. Essentially, that has conferred more information about where we are, where the mouse was, into that. In the FM1 knockout mice, we can still produce a response. It's, as I suggested, a bit smaller than the wild type, but they're really not able to enhance that synaptic strength. So they're not able to reinforce that connection. And when we quantify that in numbers, what you can see is this is the wild type mice. They can always strengthen by about 50%, and the FM1 knockout mice really can only do it about 10% of the time. And the last couple of slides I want to talk to you about is a really interesting finding we had recently where we were looking at this and coming back to this kind of neuron idea. These long-range inputs come out to this distal region of the cell, way up here in the tip of the dendrites. And lots of drugs have been trialed for Fragile X syndrome, both in clinical trials and preclinical trials, that have kind of had a logical basis in the function of the neuron across this axis, but very little target-specific regions. And just kind of to throw a few examples out, there have been clinical trials that do metonide, metformin, lovastatin, and the mGluR, phenibam, mavaclorinate, etc. And they really hit the whole cell, and they target synapses across the whole region. But one drug, baclofen, my previous research has shown has selective effects on this outer portion where these long-range inputs arrive. And we want to ask whether or not, if we gave our baclofen to the neurons in the mouse model, could we see some rescue of this kind of synaptic strengthening? So to do that, we did exactly the same experiment which I just showed you, but now we stimulate after we've applied our baclofen to these neurons. And so in wild-type mice, satisfyingly, we can see that our baclofen doesn't really affect their ability to produce this strengthening of inputs. But in the FM1 knockout mice now, what we see is that following our baclofen administration, we can see a recovery, or we can see an increase in the synaptic strength now. And when we quantify this, what was very satisfying was that we still, and this is our control data I showed just there, we see this reduction of synaptic strengthening. But when we give our baclofen to these slices, the level of synaptic strengthening now roughly equates to that of wild-type levels, indicating that potentially baclofen, although it failed at clinical trials, may have some benefit in terms of this type of memory formation. Whether or not that's the only benefit it has, or whether or not that translates to humans, we're still trying to work on, and how it affects all of the other spiking data I just talked about at the start, we're still working on actively, but it seems to suggest there might be a mechanism, at least at the circuit level, that explains why some benefit was seen with that drug. So in summary, what I've shown you is the F1 mouse model is useful to understand cellular function for Fragile X syndrome. The neurons in the hippocampus display increased activity, and this results from changes in this axonal segment region. These changes appear to be due to reduced local inputs, and these local inputs themselves are less able to compensate for new activity. With that, I want to particularly thank Max Wilson and Laura Simos de Oliveira, who did so much of this work, the other members of my lab, Peter Kine and David Wiley, for their continued support for unknown reasons, and then all of our funders. Thank you very much for listening, and I'll take